So I'm gonna just start the recording here. And hopefully it works this time too. So, so far, we've covered the basic properties of water and the biochemistry, basically its definition, as well as sort of the other important definitions like acid base and things like that. A lot of equations, as you can recall, about the entropies, enthalpies, as well as the any kinds of redox reactions and things like that. So from today, we're going to move on to the some biomacromolecules and study their sort of biochemical properties, as well as how they are involved in biochemical reactions, basically for sustaining life. So today, we're going to focus on nucleic acids. So I want to just start with the central dogma of biology that DNA makes RNA, and RNA makes protein. As you know, I mean, you pretty much like covered this a while ago in your, I don't know, middle school or high school. But basically, DNA is for the, it can replicate itself to either sustain the genetic information as well as just giving it inherit to the next generation. And through the process called the transcription, RNA is made out of DNA, and this RNA works as a sort of a mediator between the DNA and actual player, actual effectors for the any kinds of biochemical reactions. And of course, after the translation from the RNA, you get proteins that are actually doing something, actually have some functions. So today we're focusing on nucleic acids again, meaning that we're going to talk a lot about DNA and RNA and how they are replicated or generated, basically. So what are the nucleic acids? Those are basically DNAs and RNAs. And as we know, DNA is deoxynucleic acid, ribonucleic acids, and the RNA is ribonucleic acids. And there are little differences there that we're going to point out actually in a few moments. There are different functions for DNA and RNA. I basically talked about this really briefly when I explained the previous slide about the central dogma of biology. But can anyone say it again, or in your own words, what the functions of DNA is? That is correct. What about RNA? Can anyone? RNA? We've heard of it a lot because, basically mRNA vaccines and the coronavirus and things like that. How about you? Raise the hand. I saw the hand raised there. To make proteins, a stuff that can make protein, basically. You're all correct that DNA is basically genetic materials for all living organisms. So whenever we talk about some living things, we can't exclude the DNA there. It just have to be there to sustain its life and going on to the basically next generations, the progenies. And RNA primarily serves as a mediator between transcription and translation. So it is a stuff that is for making protein, but it also has its own functions as enzymes or some kind of important functional groups too that we're going to talk much later in this semester or this course. And some viruses have RNA genomes too. So it's a pretty interesting function because RNA essentially should not convey or contain the genetic materials, but it actually does, and it can be sort of reverse engineering, right? It contains the RNA and can generate the DNA later on once it gets infected, infects its host cells and can propagate, basically. When we talk about the structures of nucleic acids, it's pretty simple. It's just a linear polymer of its a subunit called the nucleotides or nucleosides. And DNA basically has a lot of these nucleotides called the deoxyribonucleotides, as opposed to RNA contains nucleotides called ribonucleotides. And the basic difference between these two is the little functional group in the sugar. Again, that we're going to talk in more depth in a minute. And the nucleotides basically have three components or three parts: nitrogenous base, pentose sugar ring. It's basically ribose, and the phosphate group here. And DNA, like I told you, there's only a small difference between DNA and RNA, which is whether this sugar ring, the pentose, is deoxyribose or just ribose. And at the physiological pH, which is around 7, every nucleotide or the residues within the big, like polymer, should carry some kind of charge there. Is it negative or positive? Can anyone talk about this or choose one? Is it negative or positive charge? Why is it negative? Because why? Because of the phosphate group. How so? I mean, I can see the phosphate group here. Exactly. So the negative charge or its property, electronegative property, basically comes from this phosphate group, because obviously you can see this negative signs here, right? Which means that it lacks an electron. So it tries to just recruit the other electrons. So it has the electronegativity there, makes this whole molecule negative. So now we're going to talk about each component: nitrogenous base, sugar, and the phosphate group one by one. So let's talk about the nitrogenous base first. And I'm going to write it as N bases a lot, on this slides and during my lecture too. Sometimes we'll call it N bases. There are like two major types. One's purine and the other is pyrimidine. And as you can see, both of them have sort of the ring kind of structure there. Purines have two fused rings, as opposed to pyrimidine has only one ring. And you must understand and know, and pretty much like I'm talking to you about memorizing the structures and the atom numbering conventions for this purine and pyrimidines. And you can see that the numberings here for purine starts from nitrogen here as 1, 2, 3 to 6, and then 7, 8, 9, as opposed to purines are starting numbering 1 from this nitrogen on the bottom here and goes the counterclockwise. Is it clockwise actually? This one's counterclockwise. So they are different in numbering. So you should note that. They are sort of a little bit different. And this is not about making you guys confused and making into some kind of exam questions, but it actually follows some rules. Can anyone actually explain this kind of numbering rules? Why they're having those kind of, looking like a vastly different kinds of numbering systems? Has anyone attended chemistry or the organic chemistry? What is it? Yeah. So it basically follows IUPAC. I mean, if you have heard of it, it's just a way to name all the chemical compounds. And as she pointed out, there are some rules there to follow. Basically, if you see some rings, and if the constituent is different than carbon, it always starts from there, like a nitrogen. So it always starts from the nitrogen there and has to be linked to the biggest functional group first, which will given as number 1. So, and also there's a double bonds and things like that. There are some couple more rules to govern this. So if you want to really understand what's going on here as numbering, you can just quickly look up the IUPAC, nomenclature there, and will should make sense to you. Let's keep talking about the nitrogenous bases, especially about the common features. So as we've pointed out, those are aromatic rings. One is the purine has the fused 2 rings, basically, as opposed to pyrimidine has just a 1, 6-membered ring. And they are aromatic rings, but there are heterocyclic structures. What does heterocyclic structure mean? What is being heterocyclic? I'm just asking this bunch of little questions because I just assume that you guys have taken the prerequisites, which are chemistry and organic chemistry. Two different rings. So there are two different rings there, which forms a heterocycle. Any other person? Alternates double and single bonds. Other people? Maybe I should make it into my exam question, which is sort of a pure chemistry, but what the heck? Exactly. So typically when you see aromatic rings or some kind of benzene kind of structure, some kind of a multiple membered kind of ring, many times you see a single atom there, like a carbon and things like that. But it's actually a mixture of different atoms there, carbon and nitrogen, right? So that's why it is called as a heterocyclic. So it doesn't have to be just the two. It can be 3 different or 4 different kinds of atoms that are involved there. And that actually confers a really important property to the nitrogenous base that we're going to talk about a little later on. And when you look at the structure, so it's all about chemistry here. So let's say that we're just sort of refreshing our old kind of knowledges here about the simple chemistry. Question. Oh, so benzene is not. That's a good point. So maybe it is a little confusing. So nitrogenous bases are heterocyclic rings, basically. But benzenes are not. They are homocyclic, right? But the prototype is that. The shape of that is there. And this is sort of a variant, derivative of the benzene ring. So benzene ring is not heterocyclic. All right? And when you look at the structure, so we're going to just take an example here, a homocyclic ring actually, which is benzene here. And when you look at the structure of this, it has alternating double bonds. And these double bonds, because all the carbons are sort of equivalent there in terms of their space, their positions and the sort of capacity to hold on to the electrons and things like that. This double bond can move to the other side of the carbon, when you look at focus on one carbon atom. And that is called as resonance. So it forms the other type of alternating double bonds here. And it is pretty much dynamic, right? It can go back and forth really quickly there, sort of making into an intermediate stage where there's a sort of double bonds here, some double bonds here, like a 50-50 chance or so. And it's called as a resonance hybrid, basically, where there's a 50-50 chance of electrons there. It's sort of almost like a sharing both of them as a half sort of double bonds there. And when you look at these carbons, each carbons on this cis-6 ring, they have sp3. Of course, they have to make the sp3 hybridization, as carbon has 4 legs or 4 arms to hold on to the single bonds. But when you focus on this C, then it actually have only like 3 bonds here, right? 1 here, 1 here, and 1 here. So it has the sp2 structure for sure. And there's another electron that is floating, basically shared between 2 carbons right next to each other, which forms the unhybridized p orbitals there. So it actually pops up or bottom, looking like this, as you know, makes the p orbitals, and they're going to be sort of delocalized from that. So it's, instead of making strong double bonds, it's going to float up and down, and then we'll sort of fuse there, making the hybrid there to form the this kind of cloud of p electrons there. So that creates the sort of pretty much like electron dense regions up and down, top and bottom of the ring, make this ring structure pretty stable. So this ring is not going to be like fold up, creased, and things like that. It's going to be pretty flat, like planar most of the time, although it can have a little shape, as you can recall, kind of boat shape or the chair shape and kind of thing, but the rotation is pretty limited there. So it's a pretty rigid structure there. And that's the sort of a property for most of the aromatic rings or the structures. But when you actually look at the purines and pyrimidines, they contain non-carbon atoms, especially nitrogens there, right? So it creates some asymmetry there, here and there, here too. And also these nitrogen atoms are basically electronegative. So it can allow the hydrogen bonding to the nascent water molecule or some other polar molecules there. And this, again, gives a really important function or property for the nucleic acid structure because it has to be interacted with the other things. It can be other nucleic acid or other water molecule or other proteins and things like that. If it is just a simply like non-polar, no charges there, then it's gonna just sit there, not really dissolved in water and just floating around, not interacting with other polar molecules. Make sense? Okay. There are a bunch of other informations here that we need to cover. So nitrogenous bases are found in both DNA and RNA, and there are majorly 5 different molecules are found in living organisms. For purines, there are adenine and guanine, as you know. And for pyrimidines, there are uracil, thymine, and cytosine. And as you may know, uracil is only found in RNAs, as opposed to thymines are only found in DNAs. These are just the major forms, so you're going to see most of these in this sort of original form. But also you can see some modifications happening too on top of those molecules. There are a couple of examples here. 5-methylcytosine is basically when you see the cytosine here, and the 5, so here's the numbering. So the C5 is basically right here. And instead of having just a hydrogen atom here, it'll have methyl group, which is a CH3. And that is a little marking for the cells, and it's called epigenetics. Basically marks that region for the either enhancing or inhibiting the transcriptional activation. So it has a critical role in gene expression. And another example is RNA modifications, where a lot of different kinds of modifications happens on tRNA and mRNA too. And it also related to the post-transcriptional regulations, which affects the transcription rates or the stability of the mRNAs and so on. Any questions so far? All right. And another property of these nitrogenous bases is that they make tautomers. Does anyone tell us what the tautomer is? Can anyone? Tautomers. Have you guys heard of tautomers? What is it then? I can hear somebody's just saying no quietly. Exactly. So it's called isomers. Isomers are basically the exact same molecules, not exactly the same, molecules containing the same contents, basically, same number of atoms there, but structured differently. And those are the same as tautomers. And tautomers especially are called for constitutional or structural isomers. And constitutional or structural, meaning that literally, meaning that the components are different, right? The isomers meaning that they're the same thing, but having the different structure. So you can see that there are different tautomers for nitrogenous bases. And that process, that going back and forth, sort of interconverting from one form to the other, is called tautomerization. And tautomers have 2 different forms, either keto form or lactam form, or eno form or lactine form. And keto form is basically containing ketone, as you can see here, as opposed to enol forms contains alcohol, which is hydroxyl group. And mostly, when you look at the actual nucleic acids and the nitrogenous bases in living organisms, you can see most of them are in keto form. And the reason is because it can form the hydrogen bonds. This is more polar, having more electronegativity or the positivity exposed. So it's all about the interaction with other molecules, right? If this enol form can interact with other molecules, it should have more proportion there, but it's not because it actually blocks this hydrogen atom blocks the electronegativity a bit of this oxygen molecule. Same thing applies to guanine here too. Now, so far we talked about the nitrogenous base. Now we're going to move on to the other part, another part, which is a sugar or pentose. So it's basically 5-carbon ring, but it also contains the oxygen here on top, as you can see. So this is also heterocyclic. And you can basically see two different kinds of pentose in nucleic acids. Deoxyribose, the official name, the chemical name would be beta D2 deoxyribose in DNA, as opposed to you can see only ribose in RNAs. And again, only the difference is that whether you have hydroxyl group in ribose or the hydrogen atom in deoxyribose at the position of C2 prime. And I'm calling is 2 prime here, right? And this is to avoid the confusion by calling the positions or numberings of nitrogenous base, where you use only numbers, right? 1, 2, C1, C2, C3, things like that, N3, something like that. As opposed to you use primes for numbering the sugar here. So C1 prime, C2 prime, C5 prime, and so on. Okay, and well, I can just skip this, but there are beta and D here. Does anyone can explain what beta mean here? So there should be alpha and beta, right? What are these? Subunits. So you think there are different subunits, alpha deoxyribose and beta deoxyribose? Can be. Any other person? Position on the ring. So alpha and beta can be, indicate the position on the ring. Here, number 2 actually indicates the position. So 2 prime. I mean, it's supposed to be 2 prime actually, but 2 is enough in this case. Beta is all about isomers. So once you have this kind of a little like bigger compounds, there's always a possibility how they're, different ways they are structured, right? If it is just a simple methane, then you won't have any structure there. It's always the same as tetrahedral structure. But once you have 2 or 3 different atoms are connected to each other by covalent bonding, then you should define its handedness or the structure there. So it's the way to do it. Beta is a specific isomer that defines it. So here, especially for this 2 prime, C2 prime, where the hydroxyl group is pointing to. So when it's going to bottom, it's going to be called as beta, as opposed to if it goes up, then it's gonna be called as alpha. And you're gonna, almost all the cases, you're gonna see only beta isomers in the living organisms. Same thing applies to D. So there are different isomers or anomers there. And these kind of isomers and anomers, I hope that you guys have learned these terms, basically. If not, then we can always revisit, because we're going to talk about this in later lectures. But the anomers basically tells the handedness. If it goes like this way or goes like that way, is it just putting the mirror in between of the two different forms, D and L forms, you can just tell them which sort of is it counterclockwise or clockwise that how atom is located and things like that. So again, we can cover that once we actually get to that. So hopefully, whenever you see some kind of IUPAC, nomenclatures or some kind of chemical names, hopefully you can understand what these means and can get some more information rather than just, okay, some kind of ribose losing oxygen. All right. So far, we talked about the different components, especially nitrogenous base and the sugar ring on the nucleotides. Now, the rest of it is basically this phosphate group. And when you just take into account of these two components, which are nitrogenous base and the pentose ring, it is called as a nucleoside with S here. And here is basically a hydroxyl group that is attached to the C5', which is exocyclic carbon. Exocyclic meaning that it's outside of this ring cycle. But this can be attached to the other phosphate groups, especially, to form the nucleotide. So when it can have 1, 2, or 3 phosphate groups, and of course, they're going to be named differently, like nucleoside. Again, nucleoside is just the nitrogenous base and the sugar. Nucleoside monophosphate will be nucleoside with containing 1 phosphate group here. Diphosphate and triphosphate is basically calling them the different number of phosphate groups that are attached to nucleoside. So you must understand the difference between nucleoside and nucleotide and never sort of mix them. So here, again, let's just go back to this region where there's a covalent bonding formed between the nitrogenous base and the sugar. It always happens at C1', and this particular bonding is called as N-glycosidic bond because N, it uses the nitrogen atom from the nitrogenous base, and it basically connects to the sort of glycose, the sugar. As opposed to here, there's another bonding, right, between the phosphate group and the pentose ring. It is called as ester bond, as you know, because it's linked by oxygen here with the single bonds on the both sides. So you should again, understand and have to identify these different kinds of bondings, as well as you should know where the bonding happens. So here I wrote out that here's a bonding happens at C1', and the nitrogenous base, it can be N9 or the N1, depending on type of nucleic acids or the nitrogenous base that you're looking at. And the phosphate group will be only bound to the 5' of the sugar. But it's not going to bind to any other reasons except for this 3', and that we're going to talk about a little later. But when you look at these just monomers, the nucleic acid nucleotides, it is always the phosphate group that are bound to the 5' position. And this phosphate group has the pKa of about 7. Can anyone recall what the pKa for water is, which is pretty neutral? What is it? 7.4. So the pH for some of the like fluid, intercellular fluid or the blood is around the 7.35 to 7.45. So it might be the pH. pKa is slightly different from that. What is it? 15.7? So it's about 14, so it's pretty close actually. So you can sort of get, make a guess that the water, which is pretty neutral, has a pKa of 14 or 15.7, maybe you're correct. I remember as 14, so. So pKa 7, meaning that the pKa is lower here. And what does that mean? What does that mean? I mean, what happens when a chemical substance or the chemical compound have lower pKa than water, a neutral state? What would that mean? I'm sorry? Water is pretty neutral, so you can take it as a sort of in between, So pKa is lower. This pKa of the phosphate group is lower than water. So it can be either acid or base, right? Is it acidic, more acidic, or more basic? Exactly. It's about giving up the proton, right? That's the definition of pKa. We learned it last lecture, right? pKa basically tells you the ease of giving up the proton, right? So it's more acidic, right? And that kind of makes sense because there are a lot of oxygens which are electronegative. So you try to just give away the protons, right? Which is hydrogen atoms in this case. That's why they are negatively charged and giving up the hydrogen atoms. And giving up the hydrogen atom pretty easily, meaning that the pKa is lower. Make sense? These are critical concepts because you're gonna get questions during your exam. All right, so we talked about the difference between nucleoside and nucleotide, and it's basically inclusion of a phosphate group or not, right? And we have, again, 5 different nitrogenous bases, 2 purines and 3 pyrimidines. And their naming is basically adenine, guanine, cytosine, uracil, and thymine for just nitrogenous bases. And when they are attached to ribose or deoxyribose, now they are nucleosides, right? Their namings are slightly different. So instead of just adenine, so now it's adenosine, guanosine, and so on. So slightly different. So you should be able to use different words accordingly, whether you're talking about just a nitrogenous base or the nucleoside. And of course, nucleotide have different name from these two. So since the phosphate group try to just have a property of giving up the proton, it's an acid, right? Because it's acidic. Its pKa is pretty low, so it can be called as acid. So adenylate acid, guanylic acid, or so. And also more commonly, they're called as adenosine monophosphate for the 1-phosphate group, or adenosine diphosphate or triphosphate, and things so on. And same thing applies, for guanine and cytosine and so on and so forth. All right, so here's a quick summary. We talked about the difference again about the nucleoside and nucleotides. It's about the phosphate group. Of course, they have different naming too. And basically, the name of the nucleotide can specify what type of nitrogen base that you're talking about, as well as the position and number of phosphates too. But the sugar itself is just the same, right, in DNA or RNA. So you don't need to name them. So an example of the nucleotide name would be adenosine 5' or just a 5 monophosphate, or cytidine 3' diphosphate, and so on. And should be able to imagine or picture what this nucleotide what type of nitrogen space this nucleotide has, and also how many phosphate groups that are attached to. Again, must be able to correctly name them. And I mean, I'm not going to ask you to draw any structure on exam, but at least you should be able to picture that. So you can tell any kinds of properties on specific regions of those nitrogen base or the nucleosides or the nucleotides. And saying nucleotide 5' monophosphate is oxymoron, basically, because the nucleotide itself contains a phosphate group, right? So it has to be called as what? What would be the proper name for this? Instead of nucleotide 5' monophosphate. Yes? What is it? Oh, you can do that. You definitely can do that. Yeah, just adenolic acid or something like that. You definitely can use the acid name. That's a possibility. What I'm getting into is that instead of using nucleotide, what should you call it? What is it? Purine or pyrimidine? You could do that. It's pretty rare to call it that way, but it's definitely possible, but not as informative as just calling it as actual, base. But I'm asking about the sort of a general naming here, nucleotide. Exactly, it should be nucleoside 5' monophosphate. And what would that mean? If I call it nucleoside 5' monophosphate, what would that mean? Is that adenine? Am I talking about adenolic acid or guanylic acid? What am I referring to if I say I have nucleoside 5' monophosphate? It's a what? Nucleoside itself does not have a phosphate group, so that's true. If I just call it as a nucleoside 5' monophosphate, it's just one word, one mouthful word. What would that mean? Am I just calling the adenolic acid? That's all? Or adenosine, basically adenosine 5' monophosphate, is that all? Or am I talking about two purines here? Right, you can definitely specify it. If I call it just a nucleoside 5' monophosphate, it pulls about everything, all 5 different kinds of nucleosides or nucleotides here. It is critical because it's one of the most often used terms in the lab. So if you have done some of lab research, participated in, and if you did some kind of PCR there, somebody might have called you should put the DNTP, NTP, something like that. And those N is the nucleotide or nucleoside in this case. So it includes all different kinds of nucleotides.
If you do call it as like adenolic acid, is there a way to specify how many phosphates there are?
Good question. What do you think? No? Why not?
Because acid just refers to the fact that it's
so is adenosine monophosphate acidic? Is that an acid? What about diphosphate, triphosphate? All of them are basically acids, right? So there is no way to specify that. So it is correct. If you just call it as adenolic acid, that means everything, right? AMP, I shouldn't call it, but adenosine monophosphate, diphosphate, and triphosphate. All right, I better introduce you the shorthand so I can use them. So N bases, nitrogenous bases are written as just the one letters here, A for adenine, G guanine, cytosine, and thymine, and uracil, basically, something like that. And also nucleotides, which contain all 3 different components. Again, it contains the sugar, nitrogenous base, and the phosphate group, are called as AMP, ADP, ATP, and so on. This is for adenosine monophosphate, adenosine diphosphate, adenosine triphosphate, and so on, and so forth for the other nitrogenous bases like a GMP, GTP, and so on. Make sense? Yeah, there are a bunch of things, terms and everything, concepts that I have to introduce for this, lecture. So, and of course, I mean, we're learning this nucleosides and nucleotides, and we discuss about their properties, not because they're just chemicals, that are there, but they have some critical meaning for living organisms. So the biological significance of these nucleotides and nucleosides will be first these 4 bullet points. First ones, they are the building blocks for DNA and RNA. So serve as these nucleosides or the nucleotides are the monomers of this DNA and RNA, which are polymers. And also, they serve as a regulator for cellular metabolisms. So they're not just simply floating there to become a DNA or RNA. They just, as they are, have some kind of function there too, especially for signaling pathways and so on. So responding to some kind of environmental cues, stress response, or developmental cues and things like that. These molecules, small molecules, are used there to convey the information, little information, like works as a switch for on and off of a certain pathway. Examples are some cyclic nucleotides like a cyclic AMP or cyclic GMP. And also, especially ATP and GTP, they can serve as an energy molecule. So they carries a chemical energy there because a phosphate group itself have a high potential energy there to be linked. So it can be degraded or break down to the ADP or AMP. I'm talking about, again, adenosine triphosphate versus adenosine diphosphate or monophosphate here. Losing the phosphate group can release a lot of energy there. So it can be used as storage for biological energy. But the base or the sugar itself do not contain any kinds of energy. And also, nucleotides and nucleosides are involved in many of oxidation reduction reactions, also called as redox reactions. And we're going to talk about these redox reactions much later of this course. And they are really critical for metabolism, generating energies or storing the energy sources and things like that. And here are the examples, like a nicotinamide adenine dinucleotide, NAD, its phosphate form NADP, or the FAD, and so on. Okay, so so far we talked about the structure of a monomer for the DNA, which is nucleotide, right? Have 3 different components there, 3 different parts there. Now it has to be linked together to make it into a polymer. And in order for them to be linked together, they uses the bond between the sugars and the phosphate group, as you can see here. And the bond that is specifically used for this linkage is called as phosphodiester bond. And it's pretty straightforward because you have a phosphate here, and here's a ester bond, here's another ester bond, right? So it's a phosphodiester bond. And it always happens between 3' C3' of the sugar to the 5' C5' of the sugar. It always, the linkage is always, happens between 5' and 3'. Make sense? And once the linkage is established, then there's a leaving group from the nucleotides, and they are called as pyrophosphate or diphosphate, basically 2 phosphate groups. And that is a process that is done by the polymerase, DNA polymerase, that build up this, get these monomers and make it into a polymer, the DNA or RNA. So since there's a 1 phosphate group is left, making the phosphodiester bond, and the leaving group is diphosphate or pyrophosphate, what would be the monomer or the precursor of this nucleic acid? NTP. Why is it so? Exactly. So it is almost all the cases NTPs are used as a monomers. ADP cannot be used, or rarely can be used under the condition that there's a specific polymerase is involved. So that's why when you do the PCR, which I'm going to explain a little later, you always need to put NTP, not NDP or NMP. All right? All right, so interesting about this nucleic acids, the DNA or RNA, is that it sets up the directionality or polarity, especially on this sugar phosphate backbone. So it's called the sugar phosphate backbone because it's sort of the backbone, right? Connected to each other, making the long thread of the nucleic acid. And this direction is always like this. So here's a 3' of the pentose, and you have a phosphate group and 5', and so on. 3', phosphate, 5'. And conventionally, in convention, the nucleic acid sequence is written from 5' to 3' direction. So when you look at the GAN structure of the nucleotides, it's not about the phosphate group or the sugar. These two are forming the backbone here of the DNA. It's not about that, but actually the nitrogenous bases that actually contain some information. Because when you look at the phosphate group or the sugar, they're exactly the same, right? Nothing's different. It's just regular things alternating or the repeating itself again and again. But the only differences are these nitrogenous bases. So again, information is in the nitrogenous bases, but not in the other parts of the nucleotides or the nucleic acids. Now we talked about the subunit, its structure and its property. Moved on to the polymer, right? One big thread, and the actual DNA doesn't exist as one big thread, as you know, but it exists in the form of a double helix. Excuse me. And there are 3 major pieces of evidences in that DNA is actually forming the double helical structure there. The first evidence is the Chargaff's rule, where when you take the DNA from any kind of species, you can match the number of adenine to the number of thymine. Same thing applies to the number of C and G too here, even though the percentage of these nucleotides are different. How would that support the idea that the DNA makes the double helix? Can anyone guess or explain? Okay, A matches T, C matches G. That's the Chargaff's rule. That's great. But how is it connected to the double helical structure? X-ray. So it's a good evidence, actually. It's a third point here, where researchers also use the X-ray diffraction pattern there, crystallography, to predict the structure there. But as you know, Watson and Crick came up with this structure, the double helix, without X-ray diffraction patterns there at all. Everything was coming from their ideas, their brain, basically, based on the other evidences here. So Chargaff is one of them. I'm just trying to see that if you can connect these, the structure and the matching of the nucleotides.
So if the number of A and the number of T have to be proportional, it shows that they're complementary base pairs, which leads to a double helical structure.
That's true. So they're complementary, A and T and C and G. But does it actually tell that that's a helical structure, double helix?
I think it's due to the fact that like the molecule itself, so like if A wanted to be or it would be both accepted for like and with that they and the percentages they were able to like tell that like these go together once the structure
That is correct. It's all about the complementary, right? It does not tell you the structure, 3D structure at all. Chargaff's rule does not tell you that DNA makes the double helix. It really doesn't. If I ask that if it does, in the exam, the answer is no, because it doesn't. It only tells you that maybe there are couplings, base pairs there, right? One big thread and the other big thread, they're matching for some reason, That's all. Same thing applies to the keto tautomer too. It only tells you that there's a base pairs there. You have to get into more details. We talked about the hydrophobic effect, hydrogen bonding, van der Waals forces, and things like that. You have to just put down all the molecules there, match them, and try to optimize the energy there. Then you can come up with that double helical structure there. Only the Chargaff rules or the keto tautomer won't tell you that. So I'm just want to let you know that these are important evidences, but they are not directly addressing the double helical structure there. Only the X-ray crystallography or the diffraction patterns can tell you that this have this kind of structure, where it's sort of going like alternating each other and sort of rolling. So when you look at the double helical structure of DNA, they are right-handed. Right-handed meaning that you can just grab it with your right hand and your fingers should pointing up. And the anti-parallel too, which means that one goes 5' to 3'. It's about the arrangement of the nucleotides, right? The other, what is it? The other thread or the other strand should go the backward from. I'll just repeat that. So one strand should go 5' to 3' here. The other should go 3' to 5' here. So the direction is different, the opposite when you look at them. So it's called antiparallel. And the nitrogenous bases are inside as opposed to the sugar phosphate backbones are outside of that. And the reason that the nitrogenous bases are inside is because they have to make the hydrogen bondings there. So I just talked about hydrogen bonding that has to be made for making base pairs. So there are, again, 4 different subunits, A, T, and C, and G in DNA. And it's going to be U instead of T for RNA. But RNA typically exists as the single-stranded. So when we talk about the double helical structure or the double-stranded nucleic acid, we're mostly talking about DNA. The bases are complementary, like we said, based on the Chargaff's rule. So it's going to be always T to A or C to G. And when you look at it closely, A to T, T to A have 2 hydrogen bondings here, as opposed to C to G have 3 hydrogen bondings. And same thing applies to A to U instead of T. Those are 2 hydrogen bondings that are involved for base pairing. So here are 2 quick questions. Which pair is stronger? Is it A to T or G to C? Because there are more hydrogen bondings. There are more energy required there. Are there going to be any advantages or disadvantages that you can think of for having a stronger bonding, stronger pairing? Harder to break. Is that an advantage or disadvantage? Disadvantage. Because?
I don't know.
It's okay. Mm-hmm. Exactly. You need to separate them, especially for the replication. Other person? Exactly. So it depends, actually, depending on your goal or the purpose, right? If you want to tear it apart of these double helix and have to do something inside, then of course that's going to work as a disadvantage, as opposed to if you actually need to break it up and do have to hold it pretty stably in a certain conditions, then it's going to work as a sort of advantage. All right, even if they are involved in different number of hydrogen bondings for these different two types of base pairs, they still share the similar dimensions, the size, the geometry of them, which makes the regular structure of DNA. So make the pretty, really nice double helical structure there. So there's no bulges, distortions, or anything, unless there are some proteins or any other chemical compounds that are involved for disrupting these hydrogen bondings. So we're talking a little bit more about this hydrogen bonding that are involved for base pairing here. And especially when you look at the guanine and cytosine interactions, your base pairing here, there are actual atoms that are involved for hydrogen bonding. Here, the numbering, I actually forgot to put it here, I think, but the numbering starts from here. So it's gonna be 1, 2, 3, 4, 5, 6. So N1, N2 is right here, and O6 is right here, are involved for hydrogen bonding. In cytosine, counting starts from here, which is attached to sugar. So 1, and then goes backward, right, clockwise. So 1, 2, 3, 4, right? So O2, N3, and 4 are involved. Do you think, so I'm not gonna just ask this question because it can be a little confusing, unless you guys have any questions. But you should be able to identify which are the donors or which are the acceptors of hydrogen bondings. And it's all about the presence of protons, right? Connected to the electronegative atom like oxygen or nitrogen. So you can imagine that this hydrogen works as a donor as opposed to nitrogen here works as an acceptor. Similar thing applies to here too, but here's a nitrogen electronegative atom that is attached to the hydrogen atom. So this works as a donor, and the oxygen here works as an acceptor. So here's an arrow for the donor and acceptor. As you can see, should be able to identify these donors and acceptors, and base pair too. So since DNA has two strands and they're antiparallel and also the complementary, they have semi-conservative properties there. So when the replication happens, each of the strands can be used for replication, meaning that they generate the complementary strands there independently. But in the end, they're gonna share the exact same sequence there because it's highly specific. And just following the central dogma of biology, the information is conveyed exactly the same without an error in theory, which is called as a collinearity. So DNA has a specific sequence, and complementary sequence will have some kind of matching sequences there, right? Exactly the same, not the same, but the matching. And then mRNAs will have a specific sequence, but it's followed basically by this DNA sequence, which is 5' to 3'. But instead of having T, you're going to have uracil, U, in mRNA. And tRNAs, which brings basically recognizes the codons and brings the amino acids, will generate the protein, basically decoding this information from mRNA. So everything is connected here precisely. Now we're going to talk about a few techniques of where you can use DNA, either detecting the DNA or manipulating the DNA. Or also, it can be applied to RNA too. The first one is called as gel electrophoresis. And have anyone ever done gel electrophoresis? Quite a lot of people, actually. So basically, it is for separating the DNA. But by what principle? Is that based on the DNA's size, length, charge, sequence, 2D, 3D structure, or the specific, some kind of modifications or anything? Charge? How come? Okay. Mm-hmm. Mm-hmm. Okay. Any other thoughts? So the answer is size. And size actually is proportional to length and charge too, right? Because you're going to have only the phosphate, sugar phosphate backbone, and they're regular, identical, right, in terms of their monomers. So that means if you have a bigger DNA, then you're going to have higher charge, negative charge, right? So it's a charge too. But when you talk about the specific charge, like from the Zephyr-Ryans and things like that, it's not, right? It's only the actual size, the charge that was coming from the size, right? So I would rather say it's size or the length. Sequence is probably not because it's already single-stranded. When you do this kind of analysis, it can be either double-stranded or single-stranded. But the sequence really doesn't do anything because it's all about the size of the pore of your gel that you're running. So based on the thickness of your gel, it will determine the size or the speed that is migrating towards to the certain direction there. 2D, 3D structure, it can be actually, because some kind of supercoiled, circular DNAs and things like that can be migrated much faster than the linearized ones. So it's basically property of any kinds of stuff that is going through the little pores. So if you have a really kind of laid out like long thread that is pretty flexible and kind of hanging on to everywhere, then it's going to take longer. As opposed to if anything's really kind of compact together, then it's going to move faster, right? So structure can be too. But basically, it's all about size. All right, I'm going to just skip this. And another important technique is molecular cloning, because the molecular cloning is about the construction, making the recombinant DNA molecules in bulk, a lot of them, for using it as some kind of other purposes, like where you need a lot of DNAs for injection, expression, or something like that. And also, it's for just maintaining your own sort of your stock, if you will, some kind of specific DNA that you want to still hold on to and just keep it and propagating and just spread it to other people. You have to have it like ready and make it into the form where it can be replicated or easily distributed. So molecular cloning is all about making new DNA fragments and generating the population of the same identical DNA molecules. So it requires the cloning vector. So it's a sort of an empty vector. It doesn't contain any information there other than just having the minimum requirement to be amplified or replicated in a certain biological system. But you have foreign DNA or DNA in interest. And what you do is just to cut this out, the region in interest, and also cut out, make it open of this cloning vector and fuse them together. And here there are two types of proteins are involved. One's called restriction enzyme and the other is ligases. And restriction enzymes are basically just cutting off anything. It can be pretty specific or non-specific. And the ligases are for fusing different fragments together. And these restriction enzymes are really important because they have specific properties that we can utilize for targeting specific regions of the DNA or do some kind of engineering there too. So restriction enzymes cuts only the double-stranded segments, but not the single-stranded ones. And it is highly sequence specific. So that specific region is called as a recognition site for restriction enzyme. And it can create either sticky end, where it has a sort of overhangs on each side after the cut, or the blunt end, where it is sort of a blunt cut, right? There's nothing overhanging at all. And there are different types, type 1, 2, 3, 4, basically, of the restriction enzymes. But type 2 is widely used because of its property to incise, to cut the right in at the recognition site. So you can control where the cut is happening. As opposed to type 1 is also have a recognition sites, but once it recognizes the sites, it cuts at random places. So it can be problematic, right, if you're targeting certain region. As opposed to restriction enzymes, the ligases are not so sequence specific. So you can just use it as a glue for anything that is already cut. Once you have your vector engineered through the molecular cloning, now you want to introduce it into your model system or any kinds of system where you can make it propagated, right? Make a lot of replications there. And the process of inserting or introducing that vector into an organism that is used for this kind of amplification is called as transformation. And the organisms that is widely used are bacteria and yeast. And there are different types of vectors called the plasmids, phosmids, BAC, and YAC, bacterial artificial chromosome, or yeast artificial chromosomes. Have you heard of any kinds of these vectors? Anything? Question or? I'm sorry? You couldn't hear? Okay. I should just put it a little closer. This is actually much better. I should do that next time. So again, transformation is about inserting or introducing the vector into an organism where you can replicate that vector or the plasmids a lot. And typically, bacteria or yeast is used, and the type of vectors, there are different types of vectors that can be used. Here are some examples: plasmids, phosmids, BAC, and YAC. Bacterial or bacteria artificial chromosome, or yeast artificial chromosome there. And this is all about the size, actually. Nothing specific there. This should have a pretty much of a common features, share the common feature, but their sizes gets bigger and bigger as you go to the later of this list. These are really important common features for cloning vectors. The first one is origin of replication. Can anyone explain what that is? Why is it needed? Origin of replication. It has to be replicated, of course. That's the purpose of doing this molecular cloning, right? But why? Why do we need origin? Anyone? Why do we need the origin of replication? I'm sorry? Start or stop? Start. Exactly. You always need a starting point for that. Okay, thank you, Zoom. I have 5 minutes. And another thing is a selection marker. Examples are antibiotic resistance, luminescent proteins, and so on. And basically, this is to tell that whether the cells you collected after the transformation are the properly transformed one containing your vectors or not. And since your vectors expressing this specific selection markers, like antibiotic resistance, in this case, this is ampicillin resistance, it'll behave that way, right? So you can use it as a selecting those cells that contain these particular vectors. And multiple cloning site or the polylinker is another common feature where it has a lot of restriction enzyme sites, recognition sites. Why do we need that? Polylinker. Can be. Anyone? You have to insert something, right? And insert the gene in interest. You have to use restriction enzymes. And you can't just cut just random places in the vector, right? And you have to have a specific recognition sites to utilize those enzymes. So this polylinker is an artificial region where just put a lot of recognition sites, different kinds of recognition sites there. So you can just choose one or two there, cut the small region out, which wouldn't affect too much on the replication capacity at all, but still can introduce new genes there by ligation. Make sense? PCR, there's another really critical tool to amplify the DNA fragments. So I'm going to quickly just go over the process here. It amplifies specific regions of DNA, and it requires a set of a primer, 2 primers, and it also they call it as the oligonucleotides. And basically, they determine the DNA fragments to be amplified because they hybridize to the start and end of the region of interest that you want to amplify. So those are pretty short DNA strands, usually 18 to 25 base pairs, but it gives the starting point, the ground basically for the DNA polymerase to go on for the elongation. And those primers should complementary to your target DNA fragments or the region. And the process consists of the 3 steps basically. And there's a repetition of the pretty much like 35 cycles or so of that 3 steps that are involved for the PCR, the polymerase chain reaction. The first step is the denaturing, where you separate your double-stranded DNA. So you just basically tear it apart to make it open, so you can do something in the DNA. So that's why you apply a really high temperature there, and it's pretty brief as 1 to 2 minutes. And the next step is annealing, where you let the primers hybridize and attach to your DNA template, the DNA fragments in interest there. So temperature is a little bit low here, and you give enough time for the primer to find the right place on the DNA. And the third step is the elongation, where DNA polymerases copies or amplifies, replicates the DNA strands there. So temperatures around the 68 to 72 degrees C. And a common one polymerase that is used is a Taq polymerase. And the time duration depends on the length of template. So of course you have to give a little bit more time when the DNA fragments is longer. We're just quickly going on the other techniques here. Another one is Sanger sequencing, where you use the same principle as PCR, but instead of amplifying this, now you're just reading this signal there using only 1 primer. So there's a 1 binding there. And on top of DNTP, deoxynucleoside triphosphate, you use a little bit of chain terminating nucleotides, which is dideoxynucleotide or DDNTP. And what it does is basically it stops the amplification once it is used. So I know that this is over time. If you can give me like 3 or 4 minutes, I can just finish it. Otherwise, you can just leave. That's fine too. I'm just covering some of other molecular techniques here that you can use for relevant to DNA. So if you know this, then you can free to leave. And so the difference between just the regular DNTP and DDNTP is that only this, where you have a hydroxyl group on the 3' instead of in the DNTP, but you have just a hydrogen ion here. So that means you're not going to be able to make the phosphodiester bond anymore in this region. So the amplification will stop immediately. So it can happen, inserted, this DD NTP can be inserted in any random places. So you can have the fragments in multiple sizes, basically. And you can do the same thing for the specifically ATP, DD ATP, DD CTP, GTP, and GTP, and just run the gel. So this shows all the possibilities where these fragments can end as either A, C, or G, and T. So basically, you can just read it from the bottom to read the whole sequence. And this is an example of actual acrylamide gel. And more advanced technique will be using the capillary with the viscous polymer, where you use the same principle, but instead of using DD-NTP, you use the DD-NTP with the fluorescent tag, and you can just read it on the go when this sample goes through the capillary, and you can just see this kind of beautiful signals from the different fluorophores. Can read the sequence from the beginning to the end. And this is actually pretty critical. So you can also analyze the nucleic acid, especially its structure, using spectroscopy. You use basically the absorbance of the UV, which is at 260 or so, using this extinction attenuation coefficient. So there's a relationship between the absorbance of the UV light and the attenuation or the extinction coefficient by here, which is a direct correlation. So that means when you have double-stranded DNA, you're going to have low absorbance. And that means you have low extinction coefficient. As opposed to when you have a single-stranded DNA that is sort of loosened up, then it absorbs more UV light, so you have a higher extinction coefficient. So by just measuring the UV absorption or looking at the extinction coefficient, you can tell the structure or how much hybridized or paired those two strands of the DNA are. And this is the last sort of important properties of the DNA. So called the hyperchromic shift or effect. What it does is basically uses the absorbance differences between different structures of DNA. So when you just have the double-stranded DNA and you raise the temperature of that, you're going to see that the UV absorbance goes higher and higher because it becomes to the single-stranded, right? But around this T endpoint, which is a melting temperature, where the half of DNA is denatured, so single-stranded, you're going to see the dramatic changes on the UV absorbance. And that is happening dramatically, so it's called as a cooperativity, where there's a fast changes on absorbance. And that means when the one nucleic acid or the nucleotide interaction base pairs is broken, then all others nearby will break in a similar time or similar manner around the melting temperature here. So again, it is called as hyperchromic shift or effect. There are 4 major factors that affect this melting temperature. One is GC contents. So of course, as you can imagine, if you have high GC contents, you have more hydrogen bonding. So you're going to have higher melting temperature. And the other way around too. Salt concentration is important too, where high salt concentration will stabilize the base pairing or the negative charges of the DNA. So you're going to also raise the melting temperature. And sequence length is important as you're going to have more hydrogen bonding. And number of gaps is also important because when you have more gaps, that means you have less hydrogen bonding. Okay, I'm going to just skip this part and just try to cover the most critical ones here. And pH and organic solvents can affect that too. And lastly, there's a Sudden blotting, which is developed by Edwin Sudden, that to detect the DNA uses the radio labeled probes there. So you have like a typical gel run that all of the DNA fragments, and you use your probes that is specifically binding to your protein, or not the protein, but the nucleotide or the DNA in interest, DNA fragment in interest. And you can detect that fragment basically by hybridizing this radio-labeled probes with your gels. And you have other blotting too, like a northern blot, which detects RNA instead of DNA, and western blotting, which detects the protein instead of DNA. So Southern is named by a person who developed this, as opposed to Northern and Western is more like named it for fun. And same thing can apply for the Eastern too. There's an Eastern blotting too, which detects the lipids and then some post-translational modifications and so on. So this is the end of the today's lecture. And I was gonna actually cover a few things here. So you guys free to leave. But there has been some questions about the exams and so on, so I just want to talk about that. And since we have a recording, so all other students who left can also, get this information. So for the exam questions, some people, was wondering what to study, what to focus, and things like that. You should focus on the what I'm actually explaining, what I'm talking about during the lectures. Of course, there are a bunch of other things in the textbook, right? And my lectures are based on textbook, and adding a lot of different questions and informations and things like this, sometimes kind of going over time, like this. But the focus on the lectures, what we talked about, the topics and so on. And I'm gonna give you some practice exams there too, have a kind of better idea what I'm gonna ask about, the difficulty of the exams and so on. So it's gonna be pretty much kind of mixture. Some things easier, some things harder. And here, I just skipped pretty quickly here, but you remember what I talked about, right? Those are the important things, as opposed to something that I skipped means something, right? And somebody asked that if I can upload the slides much earlier. So it's kind of hard because I've been basically working on these slides like pretty much like I wanted to do that much earlier, but I started early, but in the end, right before the lecture, I changed a lot, arrangements and the questions and everything. So it's hard to actually upload it pretty early. So I'll do my best to upload it, post it in the Blackboard as soon as possible, but don't expect anything coming up like a few days earlier. And some people were worried about missing exams. Again, you can miss one exam, because basically, it can be just a drop off from your final scoring. But if you have some legitimate reasons, then you should talk to me about that. Otherwise, I won't give any kinds of makeups other than just the one makeup in the end during our final period, because it's about the fairness of the other students, right? Some students, although have some issues, just to try to come and make it up and by himself or herself, as opposed to someone just comes in and I have some issues, I have some events that I have to make and then have to just go. Then that's going to be unfair. So please just try to use that one excuse and talk to me if you have any questions. All right. Thank you so much. That's all.